Hello everyone, my name is Sankalp Nigam, and today I'll be talking about the effects benzene has on the L99A mutant T4 lysozyme's 4 surface pockets. But before we delve deeper into the structural changes benzene creates on the surface pockets, let's first understand what the L99A T4 lysozyme is. First of all, T4 lysozyme is a protein that breaks down peptidoglycan in bacterial cell walls. As I mentioned earlier, the T4 lysozyme L99A mutant has 4 surface pockets, but it also has a buried cavity in which molecules can bind that can help prevent the protein from unfolding and becoming denatured. The L99A mutation is a substitution of the amino acid alanine in place for leucine, which is residue number 99. This mutation enlarges the pre-existing cavity, making it easier for molecules to bind to the cavity. To make this easier to understand, let's look at an analogy. Think of benzene as a car wanting to go to the Golden Gate Bridge. The toll booths are the surface pockets that the car, which is benzene, has to go through in order to get to the Golden Gate Bridge, which is a cavity in this case. Benzene binding to the T4 lysozyme surface pockets perhaps leads to unknown structural changes that we will be analyzing with RMSD clustering analysis. RMSD stands for root mean squared deviation and helps us measure the similarity between 2 protein conformations, or structures. On the bottom is the formula used to calculate the RMSD. Additionally, in order to make the data easier to deal with, we want to cluster our data, since there are a lot of atoms and simulation frames we need to deal with. By using a clustering software called GROMACS, I was able to find an ideal amount of clusters and was able to see the average RMSD of each cluster, which allowed me to determine the magnitude of the protein structural changes over the course of the simulation. In the bottom right corner, there's a picture of the file GROMACS gives you, which contains the number of frames in each cluster and the average RMSD of each cluster. Through RMSD clustering analysis, I was able to determine that the protein structure had an average RMSD of 0.137 nanometers, or 1.37 angstroms, meaning that the protein did not move much over the course of the simulation. Any value under 3 angstrom signifies that the protein is fairly stable. However, specific amino acids in the protein moved a considerable amount, which resulted from benzene binding to the surface pockets and may have allowed benzene to find its way to the buried cavity. To get the specific amino acids that moved, I had to align 5 protein frames from the clustering, which represents about 80% of the entire simulation, and then compared them visually. Once I saw where the major changes were happening, I clicked on those parts of the protein and found the amino acids that shifted as a result of the benzene binding. From this, I can conclude that benzene binding to the T4 lysozyme surface pockets does result in small structural changes. The amino acids movement accounted for the structural changes the pockets undergo, and although these changes were not excessive, they did occur from benzene binding to the surface pockets, where these changes allowed benzene to bind in the first place. These clusters that had benzene bound to the protein had an average RMSD that were surprisingly lower than the average RMSD of the protein, demonstrating that the major changes were not necessary in order for benzene to bind to the surface pockets, or did not occur due to the binding of benzene to the surface pockets. Lastly, I'd like to acknowledge everyone who helped me throughout my project. I would first like to thank Dr. Amaro for giving me this research opportunity at BioChemCorp and for founding the BioChemCorp program, which is an excellent way for students to get the true experiences of working in the research field. I would also like to thank all my mentors for guiding me throughout my project and for encouraging me to keep trying no matter how hard something may seem. Finally, I'd like to thank all my fellow BioChemCorp students for also helping me with some problems I faced along the way, and for always maintaining a positive working environment. Thank you all for listening, and I hope this program helps other students the way it has helped me.